Okay, so the lesson which we are going to take today, it is about the selective breeding. If you're looking for page number, so page number we have 418, right? 418 we have page, and this is not a lengthy lesson. It is a short lesson, but very interesting lesson, right? So most probably we will try to finish this lesson today, right? If you take you interest here, so this is very interesting lesson. In fact, this entire chapter is very interesting of genetic engineering, right? The first lesson we are doing here, it is about selective breeding. So as if you look to the name, right, the name of
Mister,
selective breeding indicate, yes please.
sorry, Mister, my young brother was playing, and he was
Sorry.
opening the.
Okay, it's okay, because you are not listening to us, so that's why. It's okay, it's not a big deal. Selective breeding is which process, right? Selective mean you have to select something, right? Things which you have select on the best of its best qualities, right? And breeding, we know that the production of new organism, right? The production of new things. So selective breedings, the difference among breeds of dogs are great. If you look to dogs, we have a variety of dogs, right? This is not a good example of dogs. If you look to different organism, right? Like for example, in different crops you have looked. So nowadays we have a lot of varieties of the plants. Where did these difference come from? So what is the variation? What is the difference in the organism has come from? If it is in the organism, like in the dogs, if it is in the crops, so from where this variation comes, right? Human use selective breeding to produce animal with certain desired traits. Certain desired triad, the triad which you want to be there in the organism, right? Selective breeding allow only those animals with wanted characteristic, right? So selective breeding is the breeding in which you are going to select the triad, select the characteristic. So you will get the next generation, right, with the certain triad which you want. That is called selective breeding. Selective breeding allow only those animals with wanted characteristics to produce the next generation, right? For thousands of years, we are producing new varieties of cultivated plants in nearly all domestic animals by selective breeding for a particular triads. Let's suppose you have two organisms, right? Two species you have, of two organisms of the same species you have, and both of them has some common, some best characteristic. One has two good characteristic, and another have two good characteristic. So you want to just mix both of them, and you want all the four characteristics of the coming organism to be the best, right? A Native American selective breeds choice night a wild grass, which we are saying corn, right, or maize we so called. A native central Mexico. This was a grass which was raised in the native central Mexico to produce corn, a far more productive and nutritious plant. So this plant, which are Ch15, right? This was like a plant of modern corn, right? And this is a very productive plant which provide very too much help in the nutrition, right? Corn is one of the world most important crop, right? So if you look to corn is one of the most important plants of what? Corn is one of the most important plants of the crops, right? Now, for selective breeding, we need some methodologies, right? So two ways we have for selective breeding. One we have hybridization, and one we have interbreeding or inbreeding. So hybridization is a modern way of the selective breeding, which is very efficient, right? And if you look to the inbreeding, so in inbreeding have some shortcoming, right? Disadvantages have. We are going to take it one by one. What is hybridization? Anyone know about hybridization? Anyone who know about hybridization a little bit, you can share with me, please. Yes, Abdul Malik. Do you know anything about the hybridization process? What is
What,
happening
mister?
actually in hybridization? What is hybridization?
Um, I- Mr. Aino,
Yes, please go ahead.
hybridization is ideal atomic orbitals to
No, no, no,
form.
not in that of the chemistry. The one which we are going to study here in the biology, hybridization. You are talking about the overlapping of a hybrid orbital. No, not that. The one we are studying in the plants, in the animals. What is that
Okay, I
hybridization?
don't know.
Okay, let me tell you,
Mr.
right?
Ainoui,
Okay,
it's
Abdul
a
Malik.
process of forming a double standard nucleic acid from
Uh,
two
up to
complementary
some extent,
standards of DNA or the RNA.
yes, you are correct up to some extent. Actually, hybridization is the process in which we cross two individual, right? In which we cross two individual, or we can say two organism. This process is called hybridization. An American botanist is a person who is studying botany, right? Plants, right? Luther Burbank, right? So he developed more than 800 varieties of plant using selective breeding methods, either by hybridization or by inbreeding, but he used mostly hybridization, right? One method Burbank used was hybridization. What is hybridization? Crossing dissimilar individuals to bring together the best of both organisms. If you have two dissimilar, two unlike individual, and you cross both of them, right, together, and you get its best qualities, right? And the coming generation will have the best qualities. So when you cross both of them and the next generation is coming, so the next generation will be hybrid, right? We call it what? We call it hybrid. The individual hybrid is what? Hybrid is the individual produced by such process are often harder than either the parents. So now the hybrid which we'll get, that will be harder than the parents, and they will have best qualities of the parents, right? Many of Bourbon's hybrid crosses combine the disease resistance of one plant with the food producing capacity of another plant. Now, the Luther Burbank, which property he decided to be there in his hybrid? He take two plants, let's suppose, right? Two plants, one plant is disease resistance, and another plant was high crop producing plant. So he crossed both of them. So when he crossed both of them, so he take in the hybrid the quality of the resistance to disease and the production of high capacity for the crop, for the foods. So now the hybrid which he get, so that hybrid has these two qualities. That the hybrid or the plant he get, it was resistant to many diseases and it was producing double amount of food as compared to its parents, right? So this was hybrid plant which was being produced by Luther. The result was new line of plant that had the triad farmers need to increase food production. So if you look in hybridization, so nowadays most of the plant, most of the food, most of the vegetable, and most of the things which we are getting from agriculture, they are hybrid. Because we are getting now a very good quality, right? Very good quality, and a little space or a little agricultural land can produce a lot amount of food, a lot amount of crops, right? Julie Alberta's peaches. This is a species of peaches, for example, are among the Burbank's most successful varieties. So these were peaches which was being produced by the hybridization by Burbanks, right? And this was the most successful varieties he have crossed and he have get the hybrid. Another we have inbreeding. Now here I will add one point more, that hybridization is very important for the coming life, right? For the coming time, as the population is getting increased. So the agriculture land, if population is increasing, the agriculture land will increase because we are building large buildings, houses, factories, industries. So we are reducing, we are minimizing agriculture land. So if we are minimizing the agriculture land, so we need more hybridization, more powerful hybrid plants to produce more food from less place in less time, right? So this is very important field, especially with reference to that of the agriculture and to the coming time. Next, we have inbreeding, right? To maintain desirable characteristic in a line of organism, breeders often use inbreeding, right? The continued breeding of individual with the similar characteristics. So in this inbreeding, two organisms are going to cross between, right, to continue the breeding of individual for a similar characteristic. The many breeds of dog are maintained using inbreeding. So the variety of dogs as we have, these are being maintained by using inbreeding, ensuring that the characteristic that make each breed unique are preserved. So that characteristic which make the organism or the dog unique from other, that characteristic is being preserved, right? Although inbreeding is useful in preserving certain triads, inbreeding is useful in preserving in the triad for certain triad, but it is risky. As I told you in the start, that it has some disadvantage. So it is risky. If you have one organism and you are inbreeding it time by time, so there will be the chances of gene mutation, right? Most of the member of breeds are genetically similar. They will be similar, right? Which increase the chance that cross between two individual will bring together two recessive allele for genetic defects. So if they are same and they are breeding together, so it will lead to genetic defects, right? And it will lead to a genetic mutation, which will be later on be a problem for that organism, and it will suffer from diseases, right? And another thing that there will not be too much resistance to the to the diseases also. There will be less resistance to the diseases. Okay, that is about hybridization and interbreeding. You have 2 minutes. You can just go through this the selective breeding process. If you have any question till this place, you can ask me. Then we will move to the increasing variation. Just 2 minutes. You have to go through this topic, right? These are very interesting topic. If you have any point, any question, you can ask me now. Come on, boys. You have to ask at least one question. Okay. If there is no questions, we will move ahead to the next point. Next point we have is increasing variation, right? So by increasing variation, if you look right, increasing variation or increasing variety. How do people increase genetic variation? How it is possible to increase genetic variation? You can just look down to book in page number 419 just for a while. Selective breeding would be nearly impossible without the wide variation found in the natural population of plants and animals. So selective breeding would be nearly impossible without the wide variation of plants. If there is not with the wide variation in the plants, so selective breeding is impossible then, right? But sometimes breeders want more variation than exists in the nature. But sometimes the scientists or the breeders, they want more variation. They want a more successful plants. They want more successful animals in their lab or in their research or in their field. Breeder can increase the genetic variation in a population by introducing mutation, which are the ultimate source of biological diversity, right? So if any breeder, any scientist, any researcher want to increase what? Any scientist want to increase its variation or produce a good plants or good animal, right? So he will do what? He or she will have to bring mutation in the gene. So if he bring mutation in the gene, so it will produce a good plant in the future, right? Increasing variation, you can see on the screen now. When scientists manipulate the genetic makeup of an organism, they are using biotechnology. So if they are going to bring changes in the genetic variation or the genetic makeup of any organism, so they will bring what? They will bring mutation, and they will use biotechnology for this. What is biotechnology? Biotechnology is the application of technological process, invention, or methods to the living organism. When you are introducing information, when you are using technology with the life or living organism, this is called biotechnology, right? Selective breeding is one form of biotechnology important in agriculture and medicine, which is important in what? Which is important in agriculture and medicine. But there are many other. But beside this, we have a lot of other fields of the biotechnology. But the most important work still now is done in the agriculture and in the medicine. Now, as I told you that if a breeder, if a researcher, if a scientist want to do what? Want to bring changes, variation in his individual, right? So he have to do mutation, right? So now first mutation, we have bacterial mutation. Bacteria, as we know that bacteria is very hazardous thing, very dangerous thing to human, right? But this bacteria after mutation is used for the welfare of human, for the sake of human, for the importance of human. So mutation occurs spontaneously. It is a spontaneous process, but breeders can increase the mutation rate of an organism by using radiation or chemicals. Breeder can use this by what? This bacteria by increasing the mutation rate of an organism. And how the mutation will occur in an organism? The mutation will occur in an organism with the help of radiation or chemicals. If you rewind back the previous lesson, we have studied, right? When you were studying mutation. Many mutations are harmful to the human or to the organism, but breeders can often produce a few mutants, right? But they are harmful. But breeders can produce a few mutant individual with the mutation that are useful, that are having useful characteristics and which are being useful for human and for the organism. But that mutation will not be found in the original population. For example, scientists have developed hundreds of useful mutant bacteria strains. Different bacteria strain has been mutated and been developed, right? And it is being used for what? It is being used for the sake of human use, right? With the help of radiation and chemical, they have mutated. Certain strain of oil digesting bacteria, so which are being mutated, right, are effective for cleaning up oil spills, right? So it is being effective for cleaning oil spill, and scientists are currently working to produce bacteria that can clean up radioactive substance. So now the scientists are working on what? On the mutation of the bacteria that it will clean the radioactive substance from the environment, and it will help to remove metal pollution in the environment. So that's a very good point, right? If scientists get succeeded in this point. Another point, we have the last point of this lesson, polypolarity, right? If you remember in the previous chapter, we have studied that what is polypolarity. Where there we have an extra set of chromosome in an organism. So this organism is called polypolarity. Now, polypolarity, plants and animals. So let me tell you from the start that polypolarity in the animal will be dangerous, right? It will, the animal cannot afford polypolarity, but in the plant it can be, right? Drugs that prevent the separation of chromosomes during meiosis are very useful in the plant breeding. Those drugs which prevent the separation of chromosomes during the meiosis, they are very important or very useful in the plant breeding. These drugs can produce cells that have many times the normal number of chromosomes, and those drugs will produce that cell which will have many times the number of chromosomes, right? Mean the number of chromosomes will be more polypolarity, right? Plant grow from these cells are called polypolarity. So those plants which grow from these cells which have more number of chromosomes, they are called polypolarity because they have many set of chromosomes. They have extra set of chromosomes. Polypolarity is usually fatal in animal. As I told you, animal will not survive with that of the polypolarity, right? But plants are much better in tolerating extra set of chromosome. But plant can tolerate, and even plant can get benefit from what? From the polypolarity, from the extra set of chromosome. Polypolarity can quickly reduce new species of the plant. So polypolarity will reduce new species of plant that are larger and stronger than their diploid relative. So the plants which are polypolarity, they will reproduce quickly, right? And they will be larger and they will be stronger as compared to their ancestor and as compared to their parents. Okay. A number of important crop plants, including banana, have been produced in this way. So, with the help of polyploidy, a lot of plants have been produced with this technology. Besides bananas, we have domestic oat, peanut, sugarcane, and banana. So these are the things which are being used, which are being prepared with the help of what? Which are being prepared with the help of the polyploidy. Okay. So this was all about today lesson. I just finished it in a bit hurry because you were not asking any question. So now you can go through this lesson. If you have any question, any point which is not explained and which has doubt, so you can ask me now. I'll explain it again. If you have any doubt, you can tell me. I can explain it again.